born in Edinburgh in 1916 and died in Skye on the 9th of April of this year. He's buried in Kishon, not far from the cottage where he spent the last 30 years of his life. Many thanks for joining George's 4 daughters, Muriel, Marnie, Wendy and Purdy, and their family and their friends to celebrate George's life. At this point, I'd like to thank George's daughters, the school, Neuroscience, the Department of Anatomy for the generous funding of this event, and Jane Haley and Tom Jessell for their continued support. The daughters, like the majority of the audience, are well aware of George's devotion to teaching, administration, and editing of the Cunningham textbook and manuals, and the Journal of Anatomy. Tom Jessell and I felt that we should focus on things that you don't know about George. So here we go. So this is George in his office along the corridor. Can I draw your attention to George always had the class list. You all thought you were anonymous. He has a class list up behind. He has Cunningham here that he's constantly editing, and of course editing the Journal of Anatomy. He's a great typer, and you'll see in 2 or 3 of the photographs we have, there's always a teapot. So with George, and really after 30 years in this department here, George retired and spent the next 30 years working equally hard, transforming the family's summer cottage in Kishon into an eco-friendly and productive garden. Unfortunately, his wife died in 1992 and he was left to care for himself. This is a photograph in '78. He didn't really retire till '85, but you can see the cottage. Here's the cottage, and beside it there's a little building you can see with the orange roof. This is reputed to be the dwarf's cottage. The dwarfs still lived in this cottage for many years before this one was built. You can see this lovely road here, and when Tom Jessell and I went up to see George about 4 years ago, he had cleared the snow all the way up here and out to the main road. That was about a foot deep. On other occasions when we've been there, he's cut all the grass, again, on these slopes. Eventually, he had a very productive vegetable garden here and grew an orchard with apples and many plants, which you'll see later. This is the cottage about 3 years ago. You can see it's now a very productive site rather than the barn site I've shown you earlier. George still, at this age, trimmed the hedges. You can see they're perfectly done, and you can see the beginning of a rather productive garden running up on the right. It's been fun organising this lecture, and the person that sent me this couldn't attend, but it sent me this picture of George. The health and safety they don't have in Kishon. Here's George on his own, working at really quite a modern lathe, and he's surrounded by his beach combing. His favourite hobby was beach combing, and the person that sent me this would join him to beach comb. George, in 1941, published this paper. There's a series of about 4 papers from his PhD, and this one I like a lot. This is the first paper. He was interested in a nerve called the 11th nerve. I hope you can all remember the 11th nerve. The accessory nerve. It's interesting because it supplies the sternomastoid and the trapezoid muscles, but has no sensory fibers in it. But George was never sure on this point. You can see as a motor nerve, this is nucleus, and it turns sideways, and it forms this nerve that runs all the way down, parallel to the vagus. And one of George's delights in this paper was a chap called Bruce, who was one of the original editors of Cunningham, wrote that this was the motor nucleus here of the accessory nerve. It's actually the motor nucleus of the vagus. And obviously it's worth doing a whole PhD just to... You can see George arrived in Cambridge at an interesting time, and his supervisor for this project was a chap called Donald Barron. And those of you who are electrophysiologists who remember Barron and Matthews, who recorded from the dorsal roots, and probably were amongst the first people to see presynaptic inhibition. Anyway, this is Barron's idea. The plan was to correlate, if possible, the microscopic appearance of the nervous system and its functional capacity. And you cannot read a paper of Romanes's that he doesn't tell you that he's really interested in function. And you can well see this because he was surrounded by very good electrophysiologists when he was doing his PhD. I show you this because the whole of Cambridge was really at war. People like Hodgkin had gone off to work on radar. Other people like Matthews were working on trying to increase the comfort of RAF pilots and submariners, working in the cold and working at low oxygen tensions. Now at this point I'm going to hand you over to Tom Jessell, who's going to take you into the next part of George's career. Tom.
So first of all, I just want to express my appreciation for the existence of this afternoon and my presence as part of this celebration of a great life. And in particular, I want to thank John Kelly, Jane Haley, who's hiding up there somewhere, and several generations of the Romaneses for making this possible. One of the privileges that accompanies the pursuit of science is the opportunity to do just what you want, regardless of what anybody else thinks, and along the way you hope that you will make some interesting observations or discoveries. Yet you do this in the full knowledge that you're working in the shadow of the greats who preceded you. And the problem with this is that in most cases, those greats have vanished into the mists of time and are not available for discussion or debate. So in the field that I'm interested in, the field of nerve and muscle and movement, there are many greats. There's Charles Sherrington, there's A.V. Hill, there's Edgar Adrian, and John Eccles, to name but four. And so remarkably, as a graduate student, I had the opportunity to observe, albeit at a distance, three of those four, Eccles, A.V. Hill, and Edgar Adrian, because in 1975, in Cambridge at that time, the centenary celebration of Henry Dale was created and executed, and all of these greats came out of the woodwork. Adrian from his rooms in Trinity, Hill from wherever he was, Eccles from Buffalo, wherever he was at that point. Sherrington was no longer around. But these were observations at a distance and not the sort of discussion or debate that one would have liked. And so what I want to do this afternoon is to introduce another of the motor system greats to you in the form of George Ramones, and in a sense provide a very personal reflection, scientific reflection, on why I consider Ramones great as a scientist in his enduring contributions. And I know that many of you know George much better than I do, had the benefit of listening to his lectures, maybe even in this room. But for us in New York, where we're working to understand motor systems, the influence of George Ramones' work has been truly remarkable. And George was available for discussion and debate, because when I first met George, he was sprightly 95 years old, but his powers of recollection, of intent and motivation and execution of experiment were simply, had not eroded at all in the ensuing 73 years or something like that since he got started. So this is a very necessarily truncated, superficial attempt to convey why I think that George is so important in motor systems biology. And, I've called it Horizons and Perspective, because for the last 30 or so years, what interested George in terms of horizons and perspectives is shown on this slide. So this is the view from his sitting room window looking out onto Skye, and John and I and Anne had the privilege of sort of sharing that view with him. So I'm showing you this to sort of lull you into the sense of Ramones' perspective that is so important. And so when I first knew George, this is what he looked like. This was in 2011. He was 95 years old, plus or minus. And you can see many of the attributes that I think many of you know so much better. His sternly jutting jaw here, this sense of determination, but a slight sense of sort of mischievousness is also apparent. And so I like this photograph. It was taken within, a few minutes of first encounter, and it was the beginning of this very important introduction to Ramones for me. But this story, as narrative scientific, starts considerably earlier than that, in Cambridge, where he went as an undergraduate and then stayed on as a researcher. And so this is a picture that has been provided by I don't know who actually had this bit. Oh, it was Muir, right. And so this is George in his Cambridge digs at the time, sometime in the late 1930s, already studious, already scholarly, looking around at the world. And very shortly after this time, George began to work in the anatomy department, cutting his teeth in aspects of anatomy that he later became famous for. And so the first real publication, John showed you the first publication on this slightly esoteric part of the nervous system, but the first real juicy publication focused on spinal motor neurons and their organization. And this was published in the Journal of Anatomy in January 1941. So 70 years before that, our first encounter. And I gather that the Journal of Anatomy is in now very good local hands. And so this paper reflects George's attempt, and you can sort of see that in these diagrams here, to depict individual motor groups, the so-called motor columns, or what we now call motor columels, and place them in precise order in the spinal cord of a developing animal there. And what you'll see is that this information, first writ here in 1941, is enduring in terms of providing the intrigue of how the nervous system controls movement some 70 plus years later. So this began George's interest in motor neurons and motor systems, and that continued through the 1940s to the point that in the late 1940s, in 1949, he received a Commonwealth Fund fellowship to move and investigate this problem somewhere far afield from Cambridge or Britain. And he chose to go to the United States, and he chose to go to New York, and he chose to go to Columbia University in the Department of Neurology. Now, why he actually made those decisions, since there was no one interesting there at the time, except maybe Mettler, is an interesting issue, but nevertheless, he did. And what he did then, this just shows George around that time. So on the left-hand side, you're seeing him on the top of the world, up the Empire State Building, and on the right-hand left-hand side, he's somewhere outside. I think it may have been Yosemite, yeah. And so these are two pictures of George at the time that he was in New York, performing what is the culmination of his research career in terms of mapping motor pools. And so what came out of that from the Department of Neurology in 1951 was this definitive study, the motor cell columns of the lumbosacral spinal cord of the cat. Now, it might not get your blood boiling, but if you're a motor systemologist, this is pretty important. And he provided in this one diagram, and this is a long, long paper, a description of the organization of motor neurons. So I don't know how many of you know what a motor neuron is, but it's by far the most important neuron in the entire central nervous system, because there are billions of neurons, but it's the only one that bothers to talk to something other than another central neuron. It is the final means of communication with muscle for movement. And if you think about it, movement is the entire spectrum of all animal behavior. So motor neurons are pretty important, and George recognized their importance and decided that he had to study the organization and anatomy of motor neurons. And the thing that doesn't come across from this is the degree of technical sophistication that was necessary to produce a map of this sort. These days, if you're an anatomist, you inject a tracer into a muscle, and magically it appears in the cell bodies of the neuron, and you don't have to do very much to make it happen. In those days, these methods weren't available. So George was developing methodologies that permitted him to do the experiment by cutting near the muscle, the motor nerve, and looking for the degeneration, for the histological signs of degeneration of that neuron. So this was a remarkable technical accomplishment that he was performing. And in discussing these experiments with George, not only did he have this perfect recall of why he was doing these experiments, but he peppered them with a series of anecdotes that displayed his cantankerous nature. He told me one story from his graduate days in Cambridge. His advisor was Henry Harris, the professor of anatomy, who'd been the professor of anatomy in Cambridge for a long time. He wasn't really interested in what George was doing, so George sort of honed this independently. But apparently Henry Harris had this visceral loathing of the professor of pathology, whose name I think was Henry Dean. And so at one point in his student days in 1941, Harris and Ramones were up on the roof of the anatomy department in Cambridge on fire watch duty. And then Harris noticed that Dean was walking past below, and he turned to Ramones and said, "Have you got a brick?" And maybe fortunately, George didn't have a brick, and so that animosity proceeded. But this, you know, talking to him about science, you got this world of what it was like to exist in those days, which are very different from doing science today. So along with this anecdote comes this obsession with technical detail. And so he, in parallel with studying the motor system, would publish papers like this: a new silver method for the staining of paraffin sections of the nervous system and the staining of nerve fibers. So he was developing technologies that allowed him to perform the biological experiment. And this was important at the time, because at that time in Edinburgh, in the 60s, he was developing a technical core of excellence in anatomy that was really distinguished at the time. And two of the people who probably featured most prominently in this technical distinction were Alan Muir and Alan Peters. And Peters has written of his time in Edinburgh in relation to the Ramones School of Anatomy. And I just want to quote a couple of things from Peters' reflections on George, because they give you an insight into what the world was like in those days. And they also, I want to make the point, Peters became famous because he learned electron microscopy under Alan Muir and then proceeded to pursue the ultrastructural organization of the peripheral nerve and its myelin sheaths and the central nervous system. So in a way, we have a direct link to neuroscience today with Peter here. So, you know, there are many threads that can be seen. But what Peters reflected on Ramanan is shown here in the 3 different quotes. I was struck by the fact that the Professor George Ramones was the only one who had the authority to order anything. So every request for supplies or equipment had to go through him. He also oversaw the allocation of typists and of technical staff, each of whom had their own territories and would generally do nothing until requested to do so. So it's reassuring to see that nothing has changed in Edinburgh in the ensuing time. He also pointed out that much of the rest of Romanes's time was spent in rewriting the series of textbooks on anatomy that had been initially written by the Cunninghams. And these textbooks are still in print. You look at them and they reflect the excellence of anatomical detail that Romanes became famous for. And then finally, from this, Peters became a junior faculty member in Edinburgh and developed these silver staining methods in, you know, in reference to the earlier Romanes work before going on to do this EM. So anatomy was the place, Edinburgh was the place to do anatomy at that time. And then Romanes in reflective mood. Alan Muir died very early, and Romanes wrote a series of tributes of Muir. And in reading these, you get the impression that not only was he reflecting on Muir, but perhaps in a sort of autobiographical way, he was reflecting on his own appreciation of life in Edinburgh. I just want to quote one of these. So in this Journal of Anatomy memorial, he says of Muir, he was a forthright critic of anatomical detail for its own sake, but was not prepared to compromise the essentials of his subject for the sake of curricular convenience or interdepartmental peace, though he was always willing to discuss an experiment. And I can't help thinking that that is a very good description of what life under George must have been like, let alone Alan Muir's contribution. And so you could ask yourself, where was Romanes in that world, in that time of the early 50s? Was he respected by his peers? Did people take note of him? Or was he marginalized to the trivia of irrelevance? And I just want to provide one piece of evidence that indicates that Romanes was very much at the epicenter of the world of motor control and sensory motor systems at the time that this classical paper was published. And maybe the best documentation of that comes from this Cebus Foundation Symposium on the spinal cord that was published in 1952. And if you look who was involved, Edgar Adrian, Lindor Brown, some of the greats of anatomy and physiology, Ted Bullock, Barron that John talked about, John Eccles, Lindor Brown again, Bullock, Schweitzer. So he was very much of the cutting edge of neural science. Lorenzo Deneau also appeared, the cutting edge of neural science research at that time in the 1950s. So this remarkable power of presence and reflection, of course it has, with the passage of age, some signs of perhaps, shall we say, distortion of the past or simply failure to remember what you think. And I just want to give you one example of this. So around 1952, George wrote, I think it's dangerous when you're studying the development of anything to pay too much attention to the timing. This was, you know, from the horse's mouth, if you like. And when I was talking to George in 2011, you know, some 70 years later, this is what he said. And you have to listen a little bit carefully here, and I'm going to pray that the sound is working.
Causes.
Yeah, so that we...
To me,
yeah.
I couldn't get a timing for me timing was desperately important.
Timing was desperately important. So either he'd had a major revision at that point and had seen the light, or perhaps he didn't quite remember what was going on. But in any event, you know, you forgive him that after the passage of 73 years. One or two details are wrong. But nevertheless, what comes across from the study of Romanes in his time is a truly insightful anatomist who made an important set of discoveries. And for the rest of this lecture, what I'm going to try and do is give you a perspective on George's scientific contribution through our idiosyncratic eyes in the lab in Columbia University in New York, to try and point out to you in sketch-like form why what he discovered is so important in thinking about the neural control of movement. And the first thing you might ask is, has the Romanes canon, that study in 1951, the culmination of his anatomical studies, stood the test of time? And in documentation that it not only stood the test of time, but with those primitive techniques, he nailed everything correctly. I want to show two excerpts from a paper that was performed 46 years later in 1997 by a Dutch anatomy group. And so what they did is repeat the Romanes experiment with modern techniques. And they point out that despite the fact that since Romanes' studies, much more accurate retrograde tracing methods have been developed in mammals, a complete detailed overview of the ventral horn motor neuron has never been published, because nobody had the guts or the stamina to re-examine what Romanes has been doing some 50 years before. And when you read this paper, which is itself a long paper, what you conclude from this is Romanes got everything more or less right. The muscles of the most distal joints have their motor neuron cell groups most dorsal in the ventral horn. Such an organization was already noted by Romanes. So, you know, this paper is a testament to how much with those techniques Romanes got right, that not many things in science stand the test of scrutiny some 50 years later, yet he managed to do that. So what I want to do is give you my view, briefly, of the important things that Romanes said, and then pick them apart with this modern view, with new techniques, and ask why, and why it was so important. And so the thing that Romanes knew, and other people had pointed this out before, is the basic anatomy of the motor system, the motor neurons innervating muscle. Each muscle receives input by its own dedicated set of motor neurons, and that group is called a motor pool, and you can now visualize these pools by all sorts of new fancy tracing techniques where one pool lights up red, another pool lights up green. None of this was available to Romanes at the time. But with those methods, he still described the precise position of every group of motor neurons in the mammalian spinal cord. So we're going from the fact that you have one muscle, which is innervated by its own set of motor neurons. And then you have many muscles activating a given joint. So if you have a knee or an elbow, it's not just one muscle dedicated to flexing and extending, but there are 3 or 4 muscles. And what Romanes noted is that the motor neuron pools that innervate muscles that have synergist functions are themselves grouped together into larger order hierarchies that again show this sort of spatial coherence. So this was the first insight that Romanes had. And the second insight is that this organization of motor neurons in the spinal cord pays a great deal of attention on the organization of the target limb muscles that those motor neurons are supposed to innervate. Because the motor neuron, viewed in one way, is a slave to the mechanics of the limb. The limb is going to develop, you're going to get skeletal elements, the muscles are going to attach, and they're going to have to make biomechanical sense to move the limb at the shoulder shoulder, the elbow, the wrist, or the digits. And the motor neurons, this is Romanes' true observation that we still don't understand the significance fully of in molecular terms. He showed that the motor neurons respect the axes of limb organization, the shoulder to fingertip axis, the top to bottom axis, the front to back axis. And motor neurons are arranged in the spinal cord in these groups in a way that precisely matches the position of the muscle. So I'm just going to show you one axis here, the proximal to distal muscle. So in the leg, this is the hip down to the tippy toes here. And if you look, there's this inverse relationship. The motor neurons that are found most ventrally innervate the hip, then come the knee, then come the ankle, then come the foot. And Romanes, in his 1951 paper, pointed all of this out with a remarkable clarity and precision that endures today. And when you talked to George about this, he would describe what he was thinking about at that time.
Well, you see, when I look at the developmental element of this, what I see to begin with is the groups in the most ventral part forming clumps of cells. The rest is less clear, but as you go dorsally and caudally, you come into this situation where more and more cells are clearly grouping themselves.
So as you'll see in this series of video clips, one of the other features of George is that he would never let you get a word in edgeways. Each of these videos starts with me trying to say something, and then he takes over with a monologue about something that he thought about in 1941 and hasn't let go. So what we've been doing in the lab is trying to understand these features of organization. Why do motor neurons bother to sort themselves out in these precise positions that match the register of the limb muscles they're going to innervate? And in each case, you're going to see partial answers to this because we don't understand the full significance. But I want to give you a sense of how this impacts the field today. So the first thing that you could ask is, this is a fantastic cell biological patterning problem. Motor neurons appear in the same place in the spinal cord, whether you're a frog or a human being. So you think that you're using a lot of developmental, genetic, molecular, cellular effort to put these motor neurons in a place. So the first question you can ask is, how do they get into that place? What are the cellular mechanisms that put the motor neurons that innervate your ankle muscle in the top left-hand corner of this group and another set in the bottom right-hand corner? And we've been working on this problem for the better part of a decade because I think it's a fundamental problem that applies throughout the central nervous system. So Romanes provided the first, most detailed description. And I want to tell you in a few slides the idea that one family of proteins that act as surface glues, if you like, that stick cells together, are part of this Romanes positioning principle. And the proteins are cadherins. There are a lot of them here. None of these details matter. And they are surface proteins, so they're sticking out of the cell membrane. And one protein will interact with a like protein on another cell, and they will bind together, they'll hook together and keep those cells together. And the other thing you need to know for these experiments is that these extracellular interactions are transduced into the cell where it associates with the cytoskeletal elements by two key proteins, beta-catenin and alpha-catenin. They bind to the actin cytoskeleton. So these surface cadherins are sitting on the surface of cells acting as hooks or recognition systems, and they transduce that information through the catenin proteins. So what we have discovered over the years is that each of the different Romanes motor pools has its own surface cadherin code. So these are the different pools, the names don't matter, and these are the different cadherins. And you can see that no two pools here are different, are the same. Each of them is distinguished by a different, so it's as if you had a set of names that you use to identify different people. These are the names of motor neurons writ large by these surface proteins. And so you can now do experiments these days in experimental animals like the chick or the mouse, where you can say, are these cadherins important for the sorting of motor neurons into these Ramones groups, or are they simply coincidences? And the two types of experiments that you can do are sort of loss of function experiments, where you get rid of the protein and you see if that makes a difference, or gain of function experiments where you, thank you, John, where you normalize or change the pattern of expression. And I want to give you two examples here that show that these cadherins control this tight clustering and segregation of motor pools that Ramones first described. So on the left here, we're looking at a normal embryo where three motor pools have conveniently been labeled in different colors because we know the genes that make those differences. So there's a cluster of blue motor neurons, a cluster of red motor neurons, and a cluster of green motor neurons. Now, if we get rid of the two transducing proteins, and the idea here is that no matter what's going on the cell surface, they can't talk to the inside of the cell, so the surface doesn't matter, what you start to see is that this neat segregation breaks down completely, and now salt and pepper-like blue is intermingled with red, is intermingled with green. So this type of experiment gave you one piece of evidence that cadherins, via their catenin transducing function, are essential for this Ramones positioning of motor neurons. The other type of experiment that you can do is this gain of function experiment. So you can take two motor pools, the yellow and the red, and we know their surface cadherin profile, and we know that the yellow differ from the red by the expression of only one cadherin, this MN cadherin. So you can do experiments where you can get rid of MN cadherin from the yellow, or you can add it to the red, and what you see is the same with different colors here, that there is a mixing of motor neurons that are normally precisely segregated. So those two sets of experiments, and other things that I don't have time to indicate, say that cadherins are part of the Ramones motor pool positioning program. They're not the only part, probably, and we're trying to study more of this. So the cadherins here are good at making motor neurons separate, an essential feature of the Ramones observations, and this just points out that those separations relate to the muscles that they innervate. So this gives us some insight into the first aspect, in a sense maybe the less interesting aspect of how the Ramones pool positioning is established in the first place. But the more intriguing question, I think, is why do you bother to put motor pools in these positions? What good does it do? If it's evolutionarily conserved from frog till man, the assumption is that it must be doing something. And George had thought about these issues at the time that he was doing these experiments, and he had many theories, and we debated backwards and forwards in Kishorn about these theories. He didn't like my theories. I tried to point out why some of his theories may not have, been quite right. He didn't want to hear that. And so he had a set of thoughts.
And so,
because any bundle of nerve fibers growing down the spinal cord that may have some function in relation to limb movement will hit the muscles that are most proximal first. And the ones related to the toes. Last,
which is
because that's the way they're laid out, and you have to stream into them. And however you may think these descending tracts are organized, nevertheless, some of them are going to have to stop or get held up as they go down on what is the more proximal part of the limb rather than the more distal part of the limb.
I'm not used to being reduced to monosyllabic grunts of yes. It's very disturbing, actually. So we had tested this idea that Ramones was interested in the relationship between the position of the motor neuron in the spinal cord and the target muscle that it innervated. And because we knew that the cadherins could scramble things, we could design an experiment where we scrambled up the motor neurons through manipulating cadherin signaling and asked now whether under their new positions, a motor neuron that we could recognize through other profiles of gene expression would still innervate its appropriate muscle target. And so what we found is that, in fact, no matter how you scrambled the motor neurons, an individual motor neuron defined with some other parameter still innervated its correct muscle target. So the answer was that pool positioning was not important for innervating a particular muscle target. So in the absence of any information about pool positioning and target, we began to consider the idea that maybe the positioning of pools is for the inputs that motor neurons receive, perhaps from the proprioceptive sensory neurons. So every motor act is accompanied by some form of sensory validation. The muscle is telling the central nervous system how well that motor task has been performed. And the way that it tells these motor neurons is through a set of sensory neurons called proprioceptive sensory neurons, which have been studied from the time of Sherrington and onwards. And so one question you might ask is, well, there are lots of other inputs to motor neurons. Does proprioception matter for motor control? And it turns out that you can show in humans that proprioception matters, because there are a few unfortunate individuals who have viral infections where the tropism of the virus kills those proprioceptive neurons selectively. Every other, the touch and the pain neurons are still there, the proprioceptive neurons are gone. And so you can ask in those individuals, how well do they perform a simple motor task that you take for granted, like standing up and getting out of a chair? And so I'm going to show you a video of one of the individuals with these proprioceptive lesions, Charles Fried, who's been asked under video monitoring to get out of a chair. And this is in real time, so he can move, he's not paralyzed, but you can see that he has extreme difficulty in actually doing a task that all of you, most of you, would find relatively routine. And so he has to use all sorts of different motor strategies and use his center of gravity and balance, even to get out of the chair, let alone to stand up. And for Charles Fried, this is a triumph, so you'll see the look of accomplishment on his face at having been able to achieve that task. So this is merely just to convince you that sensory input to motor neurons is important in terms of motor control. And then it raises the question of how do sensory neurons find the right motor neuron targets. So in a typical limb, in your arm or your leg, you've got some 60 or 70 different muscles that are innervated by 60 or 70 different motor neuron groups that in turn provide sensory feedback from each of those muscles. And so this is a big, complicated wiring diagram that has to be fulfilled during development. And the way that developmental biologists think about this problem is influenced by two of the giants of the field, Roger Sperry and Ramon y Cajal. And so Sperry in the 1940s and 50s pointed out that neurons might have different cell surface labels of the sort that we saw with the cadherins, and that ingrowing neurons and axons might be looking for those labels, and so the label of a neuron is important. But the Ramones observation is a reflection of the larger world that Cajal had pointed out, that neurons of a given type are always found in the same position from animal to animal within a species, and as I've implied, across species. So location of a neuron is also, in a sense, a product of the identity of that neuron. And it raises the question of whether label or location is important in determining sensory motor connectivity. And George, in the 50s, was fully aware of the relevant literature that was relating to this developmental debate. And he even wrote about it. And I just want to illustrate the breadth of his thinking about how motor systems develop from these quotes. This was written in 1952. Because it would seem that there is no definite evidence of cord reorganization from the functional point of view in animals that have had their peripheral nerves or the tendons of their muscles exchanged. He was referring to Sperry's experiments. An animal like a rat, which has, say, had the tibialis anterior and gastrocnemius change, will continue always to plantarflex at the ankle when it means to dorsiflex, and to dorsiflex when it means to plantarflex. And it will go on doing this for years. And these were the experiments that Sperry did before he moved to the visual system and essentially introduced the same idea more definitively. Eventually he got the Nobel Prize for these studies, and in 1952, when Sperry's experiments were a refined group of studies available only to a select few, George was right on top of this, analyzing and realizing the significance of this. So we have been testing, in a sense, more recently in the lab, the Ramones position idea in sensory connectivity. And the type of experiment we'd like to do is to ask the question, what is important about these motor neurons? Is it their surface labels, or is it the position that they occupy? And if it's the position that they occupy, then we might be able to do an experiment where we scramble those neurons, or we even strip them of their identities, and then ask about the way that sensory neurons hook up with these motor neuron targets. And we could expect two potential outcomes. If the connectivity was just a jumble at random, it might support the idea that neurons that have been normalized don't have identities anymore. It's the surface label that's important. But if we found that despite the loss of identity, the neuron was, you could predict the relationship between sensory input and motor neuron output, then it might mean something special and different. So we did that experiment, and maybe I won't take you through the way that we did that because it's complicated and molecular. But what we did is we stripped the motor neurons that innervate the limb muscles of all identity, and then asked, did that make a difference to the animal in terms of motor behavior? So if you do that, you take the 70 different motor neuron subtypes, you normalize them, you equate them, and you ask, in a mouse you do this, you don't do this in a human. Ask, can the mouse move even? What you see is this. So this is a mouse that it has its limb motor neurons normalized in this way. So it's not paralyzed, but it has extreme difficulties in moving. Its limbs are extended permanently in this way, and it's obviously having a difficult time walking. Not, totally unlike the human Charles Fried example that I've shown you. And we can record from the muscles and show that all sorts of things have gone wrong. These two muscles that George was talking about, the tibialis and gastrocnemius, normally fire in alternating pattern. Now they fire together, which explains why the limbs are extended, because they're stiff, because all the muscles innervating joints are active at the same time. And we could also show that you could completely scramble the motor neurons. So this is in mouse, the normal Ramones pattern. Motor neurons projecting to one muscle have a tight cluster. In the mutant, they are completely randomized here. And so what we've done with this one genetic mutation in mouse, in a sense, is revert the mammalian or the vertebrate motor system back 500 million years in evolution. So inside all of you is a prototypic early vertebrate and fish-like nervous system that's desperate to mature. And so in this one molecular stroke, we've created a situation where the spinal cord thinks it's a fish, but it's faced with mammalian limbs that demand that degree of movement. So maybe it's not surprising that motor systems are impaired in this way. So what happens to this sensory motor connectivity problem? So what happens, and I'm just showing you a summary of this, is that the sensory neurons, despite the scrambling of motor neurons, so they've got no one to talk to here in terms of being able to recognize differences in motor neurons, continue to project to the normal dorsoventral position that they would have projected to in a wild-type animal. And that result, I think, is important. That result was performed by a graduate student in the lab, Gulshan Surmeli, who's now here in Edinburgh working on a completely different set of problems, but at least closer to Ramones' home. And so what that does at a stroke, if the sensory neurons are going to grow in because of some other determinant or cue and occupy their appropriate dorsoventral tear position, then from the perspective of the motor neuron, you'd better cluster those motor neurons together and you better put them in the right position, because otherwise they don't have a chance of being innervated by the sensory neurons that are needed to complete this reflex arc. So at a stroke, this one genetic experiment provides a plausible rationale for why Ramones saw what he saw, the precise positioning and clustering of motor neurons here. And this is true not just in the spinal cord, but this turns out to be a general principle of the way that sensory inputs come into nervous systems, not just in vertebrates, but even in strange animals like flies. Because exactly the same principle operates here in the fly, where different class of sensory neurons are going to grow into their target positions, independent of the neurons there. So this is something that George intuitively, I think, understood about the way in which you establish these connections, and it provides a rationale for that. But George wasn't content in dealing with the position of cell bodies. He was extremely interested in the rest of the motor neuron. It has these long processes called dendrites, as well as the axon that innervates muscle. And George was interested in how dendrites sort themselves out. And you can tell that because while he was making us this fantastic lunch in Kishon, couldn't shut him up from talking about motor neuron dendrites. Here he is cooking and talking.
Things that happen in a given order, and where your endmates are of any growth of axons or dendrites or whatever depends on the state of development. And things have to marry so that you end up with the right things connecting.
So he would talk about it, and he would write about it. So he's talking about dendrites and the organization of dendrites, and you couldn't stop him talking about this problem. And so we, because he talked about it, knew that it must be important. And then two fellows in the lab, Nikos Balaskas, who's sitting over there, who's come to breathe in the Ramones air, and David Ng, who's back in New York, began to approach this problem of motor neuron dendrites and what role it had in connectivity. And the idea is to extend the idea that maybe this Sperry target recognition isn't the whole story, and there are lots of things that explain the specificity of innovation absent target recognition, which is a tough molecular challenge. And so what Nikos and David have done is taken the time, Ramones-like, to map the dendritic arborization of every group of relevant motor neurons in the mouse spinal cord. And what they find is summarized here, and I'll just pick a couple of examples, that some motor neurons, like the gastrocnemius, have dendrites that are radially oriented. They're found at every one of the 360 degrees of angle starting from the cell bodies, but others are much more sort of linear in organization. So this was the first interesting thing that sort of picked up on the Ramones aspect of dendrite organization. Moreover, they found that the sensory neurons that are going to grow into those different dendrites per different motor pools decide on their trajectory very early on. Now this is going to get a little complicated here, but we're mapping sensory afferents, and these are the afferents shown here that are going to innervate the foot motor neurons, and I think you can just see that they're a different set. They're going to innervate the gluteus hip motor neurons here. And so it looks like the sensory neurons have made a decision of the trajectory, the angle of approach, way before they ever get near the motor neuron or the motor neuron dendrite. And as a consequence, consequence of that, what you see are remarkably predictive patterns of innervation, as if the trajectory towards the motor neuron and the orientation of the dendrite determines where the synapses are going to form. So if you look at gluteus motor neurons, they've got a big bunch of dendrites here and another bunch of dendrites here, and all of the inputs predicted by the trajectory come down here, and there are no inputs up on that other bunch. So what we're beginning to realize here is that you can explain many aspects of sensory motor selectivity in a way that reflects the Ramones position. If position is important, all sorts of things are happening as a consequence of that position. And so for these studies, which I've really just touched upon, we have the dendritic patterning, the afferent trajectory, and then the tear targeting that I described. And all of those three things, what I think they do is they reduce the problem of molecular specificity and target recognition from having to recognize the right motor neuron pool from 70 distractors to something that is manageable in molecular terms. So the Ramones position idea, I think, has relevance throughout the central nervous system. And I just want to give you one classical example of that before turning to one last set of experiments, and then I'll stop. So if we go back to the classical homunculus, the brain, the cortex, maps in a systematic way onto the limbs. And this was first pointed out by Wilder Penfield, where different bits of the cortex, the highest center of thinking, map and pay attention. So neurons in one region are going to project to the hip, in the next region to the knee and the ankle and the toe. That looks very much like the motor neuron organization that Ramones pointed out. And so it leads to the speculation, why would you bother to do that in the cortex if you weren't going to use the same principles of position input to get the information from the cortex down to the spinal cord? So I think we have uncovered or skimmed the surface of a principle of neuronal positioning that'll have a big deal in terms of how connections are formed in the nervous system. And I just want to end with one last set of experiments that we are doing in the lab that are trying to extend George's observations on motor neurons to other types of neurons in the spinal cord that are important for movement. And these are the local circuit interneurons that don't go very far, but we know are really providing input to motor neurons that are allowing them to fire in particular patterns. They're the counterpart to the sensory feedback system. And we've spent a lot of time in the lab sorting out the molecules that define these little interneuron subtypes. And this doesn't matter, except to say that we're going to concentrate on one group of 4 main groups of interneurons that we think are involved in motor control, and ask the question, within these primary groups, how many interneuron subtypes are there? Do those interneuron subtypes Ramones-like have different settling positions? And does the position Ramones-like influence the connectivity? And so we've taken one set of inhibitory interneurons, the V1 interneurons, and asked through molecular tricks, how many different subtypes of V1 interneurons really are there? So these are all the V1 interneuron cell bodies positioned here. And what we found, this is work from Jay Bykoff in the lab, is that 19 different genes subdivide this parental population into 50 different subclasses, more or less. So if there are 4 groups of major groups of interneurons, that means we're faced with the daunting prospect of 200 molecularly defined interneuron subgroups that are all involved in motor control. And we think this is important because in a Ramones-like manner, each of these individual populations settles in a discrete position along the dorsiventral and mediolateral axis of the spinal cord. So we are now seeing the reflection of Ramones' positional principle being applied to interneurons in the spinal cord. So in a Ramones-like way, does the position of the interneuron influence its involvement in a sensory motor circuit? And Jay's experiments argue that it does, that the position of the interneuron in relation to the position of the motor neuron makes a big difference in terms of that circuit. And I just want to give you one example. So because we have genes now that mark them, we can isolate each of these little subsets and map the connectivity with motor neurons and map the sensory input connectivity. And I just want to show one example that illustrates, most people had thought before that for a group of motor neurons, the little microcircuit that's involved is the same independent of the group of motor neurons. We now know that that's not true, and that each group of motor neurons has its own designer microcircuit, which may relate to the biomechanics of the limb. If you're moving the elbow joint, you're moving it in one plane. If you're moving the wrist, you have many degrees of freedom in movement. So we think that the circuitry, again, Ramones-like, is respecting what's going on in the limb. So if we take the gluteus motor neurons, they provide an input to this little blue cell called a Renshaw cell, and it provides a feedback system. And these neurons, these Renshaw cells, also receive sensory input. But now if we go to the foot motor neurons, there are no motor neuron connections with Renshaw, there are no Renshaw connections with motor neurons, and there's no sensory input. So each group of motor neurons has its own little designer circuit, and that greatly complicates the analysis of each motor pool, but it may be very important in terms of the control of movement, where you're paying attention to what the limbs have to do. So in these brief sketches, what I've tried to introduce is the idea that with 70 plus years of Ramones' position coming from this 1951 paper, the neuroscience community can be gainfully employed, at least for the next 20 years, in trying to study the implications of the Ramones observation. So in thinking about George and what he achieved, I think many of you may agree that there was a remarkable constancy of focus in George. Whatever he was interested in at the time became, and you can see that constancy of affect here in this pair of photos separated by some great distance. So, you know, I didn't know George then, I know George at this point, but you can sort of get a retrospective view. And in discussing with George, we discussed the past a lot, but we also began to discuss the future. And George speculated on continued encounters, and I need to convey that to you. So here's a little coda from our discussions. You have to listen carefully, and the punchline is at the end.
And then link it to the descending, then the whole thing, yeah.
Or you think?
Well, I'll come back and report in 10 years' time and where we are in this, but this is, you know.
All I can say is you may have to go to hell.
Sorry. This last slide is just to indicate that George's influence and stern gaze is still influencing a large number of neuroscientists in New York, many of whom who contributed to these experiments are shown here. And we have a living representative in Nikos Balaskas there. Thank you very much, and I'll leave you.